I'm going to talk today about something that is unusual in a sense for centre talks, at least from me, which is I'm going to focus on clinical implications of what we do. And I think one of the, I suppose, kind of slightly carping criticisms that I've been subjected to over my long career in research is that the sort of stuff that I do and that in the various departments that I've worked in do is not immediately clinically relevant. And maybe it's true that it's not immediately clinically relevant, but I think it is relevant. And I'm also going to talk a little bit about something that I've never really talked about at the centre before, which is the work that we've done in the National Affective Disorders unit. I've been a sort of helper consultant in that unit. Anne Farmer has been the lead outpatient consultant, and Tony Clear has been the lead inpatient consultant for much of the time. But the work that we do is fairly interesting, and I hope you'll find that it's somewhat interesting to hear a bit about. So what I'm going to do today is talk about something which has concerned us a lot at the centre, which is the whole business of normality versus pathology. And this is becoming kind of fashionable again because there's been a lot of talk in the serious press, a lot of criticism of DSM-5 and DSM-5 being perhaps a sort of conspiracy by doctors to medicalise things that aren't really medical. We in the centre spend a lot of time talking about normality versus pathology, and I'm going to spend a bit of time talking about that. I'm going to talk about treatment, again, something that we don't discuss a lot in the centre, but it's obviously relevant to what we do in our clinics. And I'm going to talk about treatment-resistant and severe patients because the clinic that Anne and I work in, and Tony Clear and others, is a tertiary referral service, and most of the time we're dealing with treatment-resistant severe cases. And then I'd just like to finish up talking about predicting treatment response, because in my seminar last year I talked a bit about Gendep and new meds, Shittage is here in the front, and I'm just going to give an update on what's been going on with the follow-up to Gendep and to new meds. Okay, so normality versus pathology. There is, those of you who read the British Medical Journal, it's quite a good rag actually, they've been having a campaign over this year called the Too Much Medicine campaign. And this is a quote from an editorial which appeared earlier this year. Essentially it's saying that it's not, they weren't not carping about psychiatry, they talk about medicine as a whole, that across the range of things, prostate cancer, thyroid cancers, asthma, kidney disease, and they only mention in the editorial one psychiatric disorder, ADHD, that there's a lot of overdiagnosis going on. And what do we mean by that? Well, we mean that actually men of my age dying of other causes, if you do a post-mortem examination and histology on our prostates, most of us will have evidence of prostate cancer, but fortunately most of us aren't suffering from it. Now, of course, people die from prostate cancer, but the whole business of what's normal and what's disease is something which is not just relevant to psychiatry, it's relevant to medicine as a whole at the moment. Okay, so how do we define disease? And I'm going to just take as my starting point a paper by Bob Kendall. The late Bob Kendall is one of the deep thinkers, was one of the deep thinkers during the 20th century in psychiatry. And I make the PhD students here at the centre read this paper. They say, my God, what's this old guy making us do, reading a paper that was written a decade or so before we were born? But I think it's highly relevant. So it's called The Concept of Disease and Its Implications for Psychiatry. And what Kendall does in this, Kendall, by the way, was the first chair of the steering committee of the centre. What he talks about in this paper is that really, in the beginning, medicine was just concerned with symptoms, and it was only round about the time of Sydenham in the 17th century or thereabouts that people started clustering together symptoms to make syndromes, and it was really only in the 19th century that the concept of germs and lesions came along. And these were very important concepts that actually influenced concepts of disease right up until the present time. And those of you who are familiar with anti-psychiatrists, such as so-called anti-psychiatrists such as Thomas Jage, his work, when he writes about the myth of mental illness, when you boil it right down, what it consists of is a criticism that in psychiatry we don't have any lesions. But Kendall in his paper said this is a very a very concrete concept, because a lot of the time in all aspects of medicine we're dealing not just with lesions but with statistical deviation from normality. But that in itself is not completely satisfactory. We're also dealing with, when we're talking about disease, is biological disadvantage. And this is a concept that he got from an internal medicine specialist called Scadding, who was grappling with the whole concept of disease in the context of respiratory disorders. So let us just look at statistical deviation from the mean for a start. So this slide is taken from a paper that Paul Bebbington and Randy Katz and I wrote back in the 1980s. It was at a time when we were doing something that we thought was quite revolutionary for the Institute of Psychiatry. That was social psychiatrists and geneticists were actually working together. And what we were doing was actually doing a family study of depression. And we were doing something called age correction, because when you actually study family members of people with disorders like depression, you may want to know what the lifetime risk of disorder is rather than the point prevalence of disorder. So you may ask questions about whether people have ever had an episode of depression, for example. Bebbington said, this is very interesting. What would happen if you took that type of age correction approach and applied it to my epidemiological data based on a survey of Camberwell? So Bebbington had found in a previous result, a previous paper during the 1980s, that about 11% of the population of Camberwell were mildly depressed, had a point prevalence of 11% of mild depression using the present state examination. So, to cut a long story short, what happens if you take the age of onset of those cases of depression and make some assumptions about the distribution of the age of onset and apply it over a lifetime, is that in Camberwell, in the 1980s, about half of men who lived to about my age would be depressed, and about 3/4 of women. So what does it mean, a disorder that actually affects most women and at least half of men? Is it a disorder at all? Well, of course, this is mild depression, but I'm showing this just to really get across the notion that there is no, as Kendall would have put it, no point of rarity between normality and disease. All of us, in a sense, are patients. But what makes a case of depression a case? Well, there are very wide differences in population prevalence estimates. So I've given one definition, but if you look at definition from the United States, from the famous papers by people like Kessler, they give even broader point prevalences than Bebbington found in Camberwell. Also, it's been pointed out by John Rice in some interesting papers in the 1980s. John Rice, by the way, a visiting professor here, the early days of the centre. He pointed out that there's very poor stability of diagnosis of things like depression in population studies. That's to say, if you go back and ask someone about whether they've ever had depression a second time, sometimes they don't remember, and sometimes they admit to symptoms that they didn't admit to the first time you asked them. Stability and reliability are, however, much improved if you increase your stringency of diagnosis. So if you look at a higher number of symptoms and you look at impairment, which is perhaps equivalent to what Kendall was talking about in biological disadvantage, or if you look at people who've actually sought help, then you can actually get a much tighter definition of something like depression. And interestingly, the disorder becomes more biological. So in my own work, we showed that, for example, depression that is associated with help seeking is associated with a higher heritability. So the sort of concept that we have then is that there is no point of rarity between normality and disease, but what we're dealing with is a normally distributed liability to disorder. And if we're dealing with a disorder that is heritable or familial, then you will get more relatives affected when you look at the relatives of people who are affected than you find in the general population. So in other words, this liability is actually shifted to the right in this diagram. So this is the liability threshold model of disease put forward by Faulkner, not for psychiatry but for any common disease, and first applied in psychiatry by Gottesman and Shields, actually working in the genetics hut, which is used to be just a few yards over in that direction in 1967. So, and I think really this Faulkner, Gottesman, Shields type model is what we always think about within the centre when we're talking about disease. And Robert talked about this in his introductory seminar a few weeks back, really, that we're talking about disorders where there's no discontinuity, no point of rarity between normality and the actual disorder. And the crucial thing is where you put this threshold. So where should you put the threshold? Well, Kendall would have argued you put the threshold at where you get biological disadvantage. And there's no doubt that with depression, there is biological disadvantage, even if we're all on the continuum of depression. But if we're beyond a certain threshold, some of us will actually exhibit suicidal behavior. It's often quoted that for severe depression, the lifetime risk of suicide is about 15%. That comes from an old paper by Robbins and Gouzet from St. Louis, where and Anne and I sat at the feet of Robbins and Gouzet back in the early 1980s. Robbins and Gouzet were probably wrong, and if you do the stats a bit more carefully, you get figures around about 6% of lifetime risk of suicide. There's also something which we have found in our data, and others have also reported on, which is that there's an increased risk of all sorts of physical diseases associated with depression. So increased risk of so-called metabolic syndrome type diseases. These are diseases associated with increased BMI, things such as type 2 diabetes, hypercholesterolemia, heart disease, hypertension. But there's also an increase in other disorders such as asthma. This is another interesting finding from the centre. People have been concerned in psychiatric disorders about the whole phenomenon of these disorders being in part heritable, but also decreasing fecundity. I gave this talk to some clinicians the other day, and one of them said, "What do you mean by decreased fecundity?" Having less kids, that's all. Having less kids. An interesting paper by Rob Power, one of our students in the centre, who's actually off in Boston at the moment, but Rob had this interesting paper published earlier in what used to be called Archives of General Psychiatry, now called JAMA Psychiatry, showing that there might be an increased fecundity among the siblings of people who've got depression, which might be a compensatory mechanism explaining why depression persists in the population despite being, on the one hand, heritable, on the other hand, causing decreased fecundity. And lastly, another student from the centre, James Cole, has done some interesting work showing that, among other things, there is decreased hippocampal volume and white matter changes in people with depression. He's not alone in showing this. He actually then did a meta-analysis and showed that across the board there's decreased hippocampal volume. So this is not, strictly speaking, a lesion, but it's a bit more like a lesion. And you know, if Charles was, he died earlier this year, if Charles was still around, he might be perhaps persuaded that some of the disorders we deal with in psychiatry are a bit more disease-like than he would give us credit for. Okay, that's all I'm going to say about the concept of normality and disease. I'd just like to talk a little bit about the latest evidence on treatment. And I'm going to talk about outcomes from the Starde study, which we've done some secondary analysis on in the centre. I'm going to talk about something which is not about, strictly speaking, centre business, but it certainly applies to the business that we do in our outpatient clinic, which is our antidepressants effective. There have been lots of talk in the press about whether antidepressants are effective. And some people have even got very aerated about the notion that not only might antidepressants be not effective, but they might actually make you worse, might make you kill yourself. So let's examine that, and then we're going to talk about strategies in refractory cases, the sort of things that we treat in our, sort of people we treat in our outpatient clinic. Very briefly, the Starde trial was a very big NIMH-funded trial which was conducted across 40 clinical sites, very ambitious, trying to recruit 4,000 patients. It was carried out in primary care and with psychiatric outpatients. Interestingly, the primary care physicians turned out to be almost as good as the psychiatrists at treating these patients. In this big study, which was of people who had had multiple episodes of depression and therefore were regarded as quite difficult to treat, about 30% of them remitted in the first stage when treated with a drug called citalopram, which is an SSRI. They were most likely to remit if they were well-educated, employed women and white. Will mean nothing to you if you don't actually treat patients, but they gave actually very large doses of this drug, citalopram, and they actually treated them for a longer time than is normally expected. So these are interesting outcomes if you're a clinician. They then went on and they did what they called was a semi-naturalistic approach, which is if people didn't respond to the first medication, then they were switched to another medication, or they were augmented with other drugs. And interestingly, then, I won't go through this in detail, but what actually happened is that if you go right through to the end of Starde with this sort of switching or augmentation approach, overall, 70% of these quite difficult to treat patients actually got better. So it depends whether you're an optimist or a pessimist, and whether you regard this as glass almost 3/4 full or glass a third empty. You know, many people say this is a very good result, and other people say, what about the 30% of people who, after all this expensive treatment and this big effort, didn't actually get better? Well, I guess those are the sort of cases that we actually treat in our clinic. But let me just stand aside for a moment and talk about antidepressants and the arguments about antidepressants, which have been popular in the press over the past few years. The first argument is that they don't actually work. Well, the Starde study suggests that they do work if you persist. But then there's the notion that they make you suicidal. And we've got some data from the centre about both these issues. But this is not from the centre. This is a slide summarising some work which was published in 2008 and which got huge publicity in the press. It was by a professor of clinical psychology called Professor Kirsch. I've never met him, but it's actually a very good paper. It was in PLOS Medicine, and what he did was he managed to get all of the data that had been submitted to the American Food and Drugs Agency about antidepressants, including lots of unpublished data. And essentially, what he did was, rather than take just placebo drug comparisons and look at whether they were statistically significantly different, he said, let's apply a clinical threshold. So the National Institute for Clinical Excellence in England, NICE, suggests that a 3-point difference on the Hamilton Depression Rating Scale, that's between 0 and this line here, a 3-point difference is clinically significant. So Kirsch and colleagues said, let's look at which of these trials actually give clinically significant results. Most of them actually gave statistically significant placebo drug differences. And he said, well, actually, a lot of these studies don't give clinically significant results. But what you can see at a very cursory look at this graph is that if you look at severe cases, and this is initial severity at baseline along here, that actually most of the studies that focus on severe cases do have clinically significant results. So I think what the Kirsch study shows is what actually the NICE guidelines recommend, is that you only treat with drugs severe cases, and actually most of the severe cases have clinically significant improvement. So I think this study actually shows that what we're doing is perfectly sensible. What about antidepressants making you suicidal, making you want to kill yourself? Well, here are some data from the centre, from the Gendep study, which was a multi-centre European study led by us here. And this is a paper published by Nader Perud, who was a clinical fellow visiting us from Switzerland. Nader has a long-standing interest in suicidal ideation and completed suicide. And he said, let us have a look at Gendep. We had quite nice data on what are called treatment emergent and treatment worsening suicidal ideation. That's to say, suicidal ideation that you never had at the beginning of the study, or suicidal ideation that gets worse as the study goes along. And what you can see is it is a real phenomenon. People do actually show increasing suicidal ideation. These are men and these are women. The criticism, though, has been that it's the modern antidepressants, the SSRIs, and later generations of antidepressants that actually make you more suicidal. Actually, Anne and I, as you know, used to work in Wales, and there was a member of our department who's actually made a career out of demonstrating that there's something called suicidal emergent. There is treatment emergent suicidal ideation when you treat people with SSRIs. Well, it's a real phenomenon. However, the black bars are nortriptyline, which is an old-fashioned tricyclic antidepressant, and the lighter bars are escitalopram and SSRI. And in fact, you actually get significantly worse suicidal ideation with nortriptyline, an old-fashioned antidepressant, than with SSRIs. So this is not a new phenomenon. I was taught by Max Hamilton, he who created the Hamilton Depression Rating Scale, when I was a medical student up in Leeds, that in fact, if you treat people with depression, quite often their suicidal ideation initially gets worse. What happens though over the longer term is their suicidal ideation actually gets better. So these are men and women, escitalopram and nortriptyline, everybody's suicidal ideation on average improves. So you do get treatment aversion, suicidal ideation, but overall the graphs show that it actually gets better. Okay, what about refractory cases? One of the things that we have a luxury of in our clinic, which is the National Affective Disorders unit, is that we have up to 2 hours to spend with patients. Actually, if we don't have a junior doctor there and it's just a consultant, we actually spend less than 2 hours because we can whip through it more quickly. But we've got the luxury of that long time to reconsider the diagnosis. So I sometimes give a version of this talk to clinicians, and they have in other centres maybe half an hour to see a new case. So one of the things we do is we're able to reconsider the diagnosis. And quite often you find that people haven't responded to medication that they've been given or to psychological treatment they've been given because the diagnosis is incorrect. I'm not going to bore you with lots of anecdotes, but let me just tell you about a young medical graduate that I saw a few weeks ago. She came all the way from Belfast to see me. I was quite flattered when patients come from such a long way, but often all we do is just reconsider the diagnosis. This young woman had her first breakdown in her final year of medical school, and she was diagnosed as having depression. She was treated with an SSRI, and she responded sufficiently well to actually go back and qualify as a doctor, pass her finals. However, when she persisted with being treated with this SSRI, she became extremely irritable, started cutting her wrists, and got relabeled not as depression but as having borderline personality disorder. Fortunately, she had a very astute GP, and one day this young woman came into the GP's surgery, as the GP said to me in her letter, high as a kite. And if you go back and took a history of this young woman, she actually had a classic episode of mania before her episode of depression. So one of the things we know clinically is that if you treat people with bipolar disorder with SSRIs, quite often they get irritable and hostile and start doing things like cutting their wrists. This woman actually did much better once she was taken off SSRIs and put on a mood stabilizer. The other thing we consider when we see patients in our clinic is whether to switch drugs or whether to augment. And I now, for one, am actually quite influenced by some of the things that have been found in Starde, and by the way, influenced by the Maudsley guidelines, which Shitty's helped to write, actually. So I now use things like buspirone more than I ever did before, but before Starde showed that they're really quite useful. We always have also the luxury in our clinic of working very closely with clinical psychologists. So we're able to offer psychological assessment if it's appropriate and acceptable to the patient. Amazingly, around the UK, despite all the efforts of David Clark and the introduction of IAPT, if you talk to clinicians around the country, psychological treatment is not often, well, quite often is unavailable, but we can offer it here. We still, for our inpatients, use ECT. Some young people find this quite shocking, but the trial evidence shows that ECT is actually the most effective treatment for depression. Far better than any of the medications that we have, far better than psychological treatment. Indeed, the people we give ECT to are just not amenable to psychological treatment. So we can often get quite dramatic effects. It's a short-term thing, but it does work extraordinarily well and can be life-saving. And we often we offer some other non-drug physical treatments. Let me just briefly mention those. We don't offer vagal nerve stimulation, but this is an interesting treatment which is now sometimes done for people with depression. My reading of the literature is that it has a modest effect in depression, but I've seen 1 or 2 cases where it's had a dramatically good effect, and it's actually a standard treatment for refractory epilepsy. The cases that I've seen who've responded who had depression also had epilepsy. We have, Sayan Pharma and I and others, Mr. Ashkan, our neurosurgeon colleague across the street, have done a small study of deep brain stimulation. Now, this is kind of like a modern alternative to what used to be called psychosurgery or neurosurgery for mental illness. The notion is that in severe depression, there is overactivity in some parts of the brain, particularly the subgenual cingulate gyrus and the amygdala. And the idea is you might be able to tone this down by deep brain stimulation. Now, deep brain stimulation is a technique which works extraordinarily well in some cases of Parkinsonism and in some cases of movement disorder. It can be absolutely dramatically improving. There are a few cases literally across the world of people with refractory depression who've responded dramatically well to deep brain stimulation. We've done a small study of this, just 4 cases. That we may be doing some more, but it's a sort of treatment that we're looking to the future. It may be very valuable. Something which is actually, many of you may not have heard of this, but there's something called transcranial magnetic stimulation, which has also had a somewhat mixed press because the trials so far have not shown that it's terribly effective. But there's some evidence emerging now that what's been wrong is the dose of transcranial magnetic stimulation that's been given. So there is a kit that's available only in the United States and actually in Israel. And Anne and I've seen it demonstrated. Some other people who were at the CINP in the springtime may have seen, I think Cities might have saw it as well. So there's a dramatic kit now, which I think is worth looking at. But something else that we're actually beginning to think about is trigeminal nerve stimulation. So this is a non-invasive technique. There's some evidence from UCLA that it actually is helpful in epilepsy. So the notion that vagal nerve stimulation might work in affective disorders comes from analogies with epilepsy. So we may be doing a little study of trigeminal nerve stimulation. And then lastly, light therapy. Seasonal affective disorder is a term that we don't like to use. We prefer to call it affective disorder with a seasonal pattern. But whatever, one of the things that happens seasonally around about this time of year, it hasn't happened yet, is that Anne Farmer appears on the One Show or something like that, talking about light therapy and seasonal affective disorder. And it very impresses our children's friends when your mum's appeared on the One Show. Anyway, but light therapy actually is effective. You've got to give enough of it. You've got to give at least 5,000 lux, and you've got to give it for long enough, at least half an hour. But strangely, it's not available in the NHS, but it's now quite affordable. You can go and buy a light box from Boots. And there's actually some evidence that even in non-seasonal types of affective disorder, light therapy can be effective. Okay, I'm going to finish up really talking about personalised psychiatry and predicting response. And this has been a long saga for me. Quite a few years back now, when Kathy Aitchison joined us at the centre, we decided we would try and get some grant money to get into personalised medicine and apply genetic as well as clinical approaches. And we managed to get funding from the EU for this study called Gendep, which was our multi-centre study led here by us at the centre. So the idea was to do pharmacogenetics, and doing pharmacogenetics, you can either study on you can actually either focus on pharmacodynamics, which is essentially looking at candidate genes, and if you like, pharmacodynamics is about what drugs do to the body, but you can also focus on what the body does to drugs, which is pharmacokinetics, which is really, how really looking at how genes might affect drug metabolism. It wasn't possible, believe it or not, when we started Gendep to do genome-wide association studies, but those of you just joining the centre will hear lots and lots more about genome-wide association studies. Just come back from Boston, and the latest results from the Psychiatric Genome-wide Consortium were discussed there. So astonishing things are happening. But we can also do gene expression studies. I'll just touch a little bit on those. Going to touch also a little bit on translation from animal models, and within Gendep, that's what Leo Shelkwick's group were doing here. And then I'm going to get back to clinical profiling. Okay, so large antidepressant studies on which there is clinical and genetic data available are actually small in number. So there are only really, before new meds came along, only really 3 studies. There was our Gendep study, which was the EU-funded multi-site study. There was Starde, which I've mentioned to you already, the big American trial, not to set up at all as a pharmacogenetic study, but they did have the foresight to collect blood and extract DNA, and then when GWAS, Genomewide association, was available, they did a GWAS. Then there's a small study from Munich called the Munich Antidepressant Response Study, MARS. We have the good fortune to be involved with Shitij and others to have a work package within his big European grant, NewMeds, and under that we were able to get data from Glyn Lewis's GenPod MRC funded study and from our own Gendep study, and we were able to, because this is a collaboration with industry, get samples from industry and put together what now is actually the biggest collection of people treated with SSRIs and to do a GWAS on them. Why would you bother to do pharmacogenetics on antidepressants? Well, there are limited data from older family studies suggesting that response to antidepressants is somewhat familial. These data, though, don't stand up to very close scrutiny. They probably wouldn't get published nowadays. But no, they're suggestive that there is a familial component. Then there's a priori reasoning, which actually is perhaps rather better than these early, rather poorly designed family studies. A priori reasoning goes like this. How you respond to medication depends on pharmacodynamics and pharmacokinetics and underlying dynamics, which is drug targets, and underlying kinetics, which is drug metabolism. There are mechanisms which are underlain by genes. So the a priori reasoning is that there ought to be a genetic mechanism. But it's only very recently from a paper published from this centre that we actually have hard evidence that there is heritability of response to medication. So this is a paper by Catherine Tansey, who sadly has now left us and gone to Cardiff. This is a study using this thing called Genome-Wide Complex Trait Analysis, GCTA. So we were able to use data from our Genome-Wide Association study to show that response to antidepressants was actually heritable, 42% heritable, that's quite substantial. SNP heritability, so-called single nucleotide polymorphism heritability, usually underestimates heritability. So probably the real heritability of response to antidepressants is markedly more than 40%. But as I say, astonishingly, although lots of people have been doing pharmacogenetics, this is the first time it's actually been shown that response to antidepressants is heritable. So that's the good news from our Genome-Wide Association studies. What about Gendep itself? Well, most of you have seen this slide before. It just shows that at least in most of the data that we analysed in Gendep consisted of 811 patients, and we allocated them to either S-citalopram, which is an SSRI, or nortriptyline, which is an old-fashioned tricyclic. Why nortriptyline? Because it's pretty specific as a noradrenaline reuptake inhibitor. Why S-citalopram? Well, it's the most specific of the SSRIs, and these two drugs are metabolized slightly differently, so we could actually compare the pharmacokinetics as well as the pharmacodynamics. So one of the first things we did was to begin to look at candidate genes. So this is a kind of favorite gene within the center, or at least a favorite polymorphism within the center. It's the length polymorphism in the serotonin promoter. As you know, there's a long and a short form, and interestingly, there is a difference in response depending on your genotype at this particular polymorphism. And the difference in response is that you respond better to SSRIs if you have the heterozygote LS form or the LL homozygote form, and you respond less well if you have the SS genotype, the short genotype. So we published this in 2009. Is it useful clinically? I'm afraid not. It's a very tiny difference. It's statistically significant difference, but it's a tiny effect. It's not useful clinically. What about other candidate genes? When we started Gendep, all we could do was candidate genes. GWAS was not actually possible. And overall, what we were able to show is that by and large, this slide is a rather messy one, but by and large what it shows is, it's from a paper by Rudolf Uhr, is that essentially response to nortriptyline, which is the noradrenergic drug, is by and large predicted by adrenergic genes around here, and that response to the serotonergic drug, escitalopram, is by and large predicted by serotonergic genes. Actually, none of these, if you correct for multiple testing, remains significant. So it's a pattern which is the pattern we expect, but again, small effects and not clinically useful. What about side effects? Well, these are data from a paper by Karen Hodgson, another centre student, also cavorting off in Boston at the moment at the American Society of Human Genetics. But she's got this paper in press, which is quite interesting. So what Karen did was to divide side effects up into groupings according to predicted pharmacological mechanisms. And essentially what she showed is that serotonergic outcomes are somewhat predicted by a serotonergic gene, 5-HT2C. So what are serotonergic outcomes? Well, if you take enough of escitalopram, it'll make you sick, it'll give you a headache, and it'll give you diarrhea. But it'll do it more if you've actually got this particular polymorphism. But there wasn't evidence of genetic associations with other pharmacological groupings, adrenergic or cholinergic side effects. So we then went on to do something that we could do now because it was made available by the technology, which is to do genome-wide association. So many of you have seen this slide before. It's pinched from Affymetrix, and it's just to show that you do genome-wide association by using a device such as an Affymetrix chip, and really you're looking at polymorphisms spread throughout the entire genome, single nucleotide polymorphisms, and actually now we tend to measure them using Illumina rather than Affymetrix. So what about genome-wide association from Gendep? Well, we did, and this is a so-called Manhattan plot from its, it doesn't look like Manhattan here, but they call it a Manhattan plot because it's fancied resemblance to a skyline. We did actually find something that was genome-wide significant in Gendep, and this was published in a paper with Rudolph, again, as the lead author in 2010 in the American Journal of Psychiatry. We got one genome-wide hit, but it was with nortriptyline. And sadly, no one else in the world has got a large series of patients treated with nortriptyline. So this has never been replicated. So it may be true, but I don't know if it's true because it hasn't been replicated yet. It was genome-wide significant. We got some other hits which were almost genome-wide significant with escitalopram, and those have not actually been replicated elsewhere. So what's happened with GWAS so far is this. No single nucleotide polymorphism, no single SNP, no single nucleotide polymorphism explains a clinically significant proportion of variance. And this is in a paper which came out in Biological Psychiatry, again with Catherine Tansey as the lead author, just about a year ago. We had great difficulty getting this actually published in a leading journal. We did get it into Biological Psychiatry because it's a very unpopular finding, because people have spent a lot of effort thinking about pharmacogenetics and designing the studies and funding them. And it turns out that as a result of all this GWAS, apart from possibly nortriptyline, which no one's able to replicate because they haven't got the samples, no single SNP explains a significant proportion of variance in thousands of patients treated with SSRIs. And NewMeds is the biggest sample, but we were able to do a combined analysis. That's to say, colleagues, Roy Perlis and others at Harvard were able, and Rudolf Uhr, were able to do a combined analysis of Starde, the Mars, that's the Munich sample, and Gendep. We actually found one genome-wide hit that was associated with early response. But again, it was a small effect and probably not clinically useful on its own. Too bad. We have got somewhere possibly further by taking slightly different approaches. So this is work, paper by Karim Malki. It's from Leo Shalkowicz's group, working on Gendep. So what they did is they treated animals, mice, with the same medications that we treated human beings with, and they found that there were 17 differentially expressed drugs with nortriptyline or escitalopram. Expressed genes, rather, with nortriptyline or escitalopram. And they then went back, Karim and Leo went back and looked at our genome-wide data to see if any of these differentially expressed genes mapped onto hits genome-wide. Not looking at genome-wide significance now, looking at nominal significance. So what they found was a gene that I'd never heard of before, called protein phosphatase kinase 1, was significantly associated with response to nortriptyline in humans. So this is translating from mice to humans. So this actually withstood response to multiple corrections and was also published in Biological Psychiatry 2 years ago. Look what it was with. It's with nortriptyline. No one has got a nortriptyline sample to replicate this, I'm afraid. So we may have 2 hits with nortriptyline, an old-fashioned drug, one on Genomewide and one based on translation from animal findings to humans, but they're with nortriptyline. So someone's got to do a big study of nortriptyline, I'm afraid. The other approach that may also be more promising than straight genome-wide is actually looking again at gene expression, this time not in animals but in human beings. And this is work from Carmine Perianti's group with our Gendep patients, showing that higher baseline mRNA levels of these species that are to do with inflammation. So this is interleukin. These other are inflammatory, pro-inflammatory cytokine type genes. Predict poor response, and again interleukin 6 and this thing, which is a sort of chaperone protein that is important in cortisol mechanisms, and these growth factors also predict response to antidepressants. So again, these findings need replicating, but they look quite promising. Okay, so the upside and the downside then of all of this is that our genome-wide association has enabled us to show that there is a substantial SNP heritability. So in other words, there is something going on there which is genetic in response to antidepressants, but no single SNP actually explains a clinically significant proportion of variance. What about pharmacokinetics? Well, this is a paper which we think is going to be published soon. It's in its second round of revision, again by Karen Hodgson, a centre student. What Karen did was to look at genotypes in something called the cytochrome P450 system. So the cytochrome P450 system is important in metabolizing antidepressants and a whole load of other drugs. And I mentioned that there's a slightly different way in which nortriptyline is metabolized. CYP2D6 is important from the way that SSRIs such as escitalopram are metabolized, where CYP2C19 is important. But essentially what Karen and others were able to show is that your cytochrome genotype does actually predict drug levels when you look at drugs or metabolites in the blood. Drug levels actually, to some extent, predict treatment response, but overall, it's not a very good thing because the treatment response prediction from drug levels is not great. When Anne saw these results, she said, "I told you so." Clinicians know what they're doing when they actually altered drug levels, and they were able to alter drug levels in the study. And unfortunately, although cytochrome P450 predicts blood levels, it doesn't actually predict treatment response either. So another perhaps disappointing finding. There are other findings though which I can't show you today because the data are still being analysed, which suggest that actually cytochrome P450 genotypes are quite good at predicting side effects. So they may actually be useful after all. So in talking about Starde and other studies, we have talked about treatment responders versus non-responders. And people often do this in studies, not just when they're talking about drugs, but when they're talking about treatment response to psychological treatments. Actually, in the real world, in real life, you don't actually get one trajectory when you get response to antidepressants. You get multiple trajectories, so that in Gendep, where we had 811 patients, there were 811 different responses. So Rudolph did some interesting work. Rudolph, we were trying to boil these down to different trajectories, and actually you can boil it down to about 8 or 9. But it looks very messy. So in the real world, there is not just a lack of point of rarity between depression and normality. There's a lack of any difference really overall between responders and non-responders. It's a bit of a mess like this. So we tried to make some sense of this mess, and I'm just going to spend the last few minutes talking about clinical features. Because the interesting thing is that if you take depression, which we think of as one thing, and you then do something called confirmatory factor analysis, and you ask confirmatory factor analysis, can I confirm that depression is one thing? The answer comes back, no, it's at least 3 things. And the 3 things, and this is another paper by Rudolph, one of the first papers we published from Gendep, where we didn't actually have any of the genetic data back in to analyse. So we were just analysing the clinical data. We showed that if you measure depression using the Montgomery-Asberg Depression Rating Scale, the Hamilton Depression Rating Scale, and the Beck Depression Inventory, turns out depression is at least 3 things. It's observed mood, which consists of these symptoms here. It is cognitive effects, consisting of all of these things, and what we called neurovegetative effects, which some psychiatrists call biological components of depression. So sleep disturbance, appetite disturbance, and so on. And interestingly, we went on to look at what happens when you apply this sort of 3-thing concept of depression to treatment response. And what we found was that observed mood and cognitive symptoms improved more with escitalopram, and with nortriptyline, the biological symptoms improved more. We thought was an interesting result. It was another thing that some clinicians say, I could have predicted this in the first place. But anyway, here we've actually shown it perhaps in a large sample, perhaps more scientifically than the anecdotal way that some of our clinicians said they could show this anyway. So if you can go on, though, with this type of multivariate statistical approach and show that not only are there 3 big factors underlying depression, but you can also show that there are multiple facets. And we were interested to see whether these facets predicted treatment response. And it turns out that one of these facets, which we called interest activity, actually is a very strong predictor of treatment response. So we looked at this thing called interest and activity, which is really about what psychiatrists call anhedonia, lack of interest, lack of activity, lack of get up and go. And many of those things get severely impaired in depression. But we were able to show that this was also associated with lack of treatment response. So this has a nice distribution, this facet, interest, activity. This is it in Gendep, and this is, we're able to collaborate with our American colleagues to look at Starde. And this is what happens when you have outcome, which is that there is a strong relationship between lack of interest and activity and response to medication, both in Gendep and in Starde. Okay, so where do we go next? So interest and activity, a nice clinical measure. It actually does a lot better than all these fancy, expensive genetic measures, unfortunately. But we're not going to be daunted by this. So we're going to go on and do methylome analysis. That's the epigenetic type work. There's still some more expression profiling work in progress. So looking at the entire transcriptome, not the entire genome, but the entire transcriptome. And we're hoping to do integrative analyses. I'll just mention that right at the end. So in fact, summing it all up, clinical measures do rather well. Cognitive symptoms and observed mood therefore predict improvement with escitalopram. Biological symptoms predict improvement with nortriptyline. And this interest activity facet, which is all about indecisiveness and anhedonia, actually predicts poor outcome in 2 big studies, not just in Gendep, but in Starde. So where do we go from here? Well, I've talked about pharmacogenetics and really pharmacodynamics and pharmacokinetics. Been somewhat disappointing because no single genetic measure has got good clinical utility. And that is shown also from genome-wide association studies. Gene expression profiles perhaps look slightly more promising, but we need to sort of follow up on that. And translation from animal models so far looks actually quite good, but with nortriptyline, and as I said, no one in the world has got a sample that is big enough to perhaps prove what we've said so far is actually correct. And we're going to also persist with looking at clinical profiles. And making sense of all of this is really quite difficult for mankind. So we're going to rely on machines to try and help us do machine learning and make sense of all of this. If you have been listening, thank you very much.